Underneath the cloud of cells, the egg itself is encased in a thick protein shell called the zona. To fertilize the egg, the sperm must break through the zona, but even the strongest can't do it by brute force alone. The egg demands a proper introduction. Proteins protruding from the sperm's cap must hook up precisely with a set of proteins on the egg's surface. If they match, the sperm is held fast and undergoes a dramatic transformation. It sheds its outer coating, releasing powerful enzymes that dissolve a hole in the zona, allowing the sperm to push its way through. The final hurdle passed, the sperm still does not thrust its way into the egg itself. Rather, the membranes of the two cells fuse, and the egg draws the entire contents of the sperm inside. Ready or not, once sperm and egg get together, they have their own agenda: to create a viable embryo. Their chances aren't great. It's estimated that more than 50% of all fertilized eggs fail to develop. If it's going to survive, the egg has a lot of work to do. First, it orders the zona to lock out all other sperm. And then the egg must finish meiosis, expelling half of its chromosomes into this tiny pouch called a polar body. With the door closed behind it, the single sperm already inside releases its precious cargo. The sperm's 23 chromosomes stretch out in the roomy, welcoming egg. The chromosomes of sperm and egg approach each other, and then the cell divides. Since the moment the sperm entered the egg, 24 hours have passed. All this time, the fertilized egg is moving down the fallopian tube toward the uterus. Every few hours, the cells divide. 4, 8, 16, gradually creating the building blocks needed to construct an embryo. On rare occasions, the tiny cluster of cells splits into two groups and creates two embryos, identical twins. But most of the time, the cells stick together. They must complete just the right number of cell divisions before they arrive in the uterus, about 5 days after fertilization. What started as a large single cell has divided into just over 100 much smaller cells. But they're still trapped within the hard shell of the zona. Now called a blastocyst, the bundle of cells must do two things to survive: break out of the zona and find a source of nourishment. At the beginning of the 6th day, it orchestrates an escape. It releases an enzyme that eats through the zona, and the ball of cells squeezes out. Free at last, the blastocyst lands on the blood-rich lining of the mother's uterus. It has just passed one hurdle, but is immediately presented with another. For in fact, it is now in very grave danger. Stripped of its protective coating, the blastocyst could be attacked by the mother's immune system as a foreign invader. White blood cells would swarm in to devour it. In its own self-defense, the ball of cells produces several chemicals that suppress the mother's immune system inside the uterus, in effect convincing the mother to treat it like a welcome guest. Then it is free to get to work. Searching for food and oxygen, cells from the blastocyst reach down and burrow into the surrounding tissue. Eventually, they pull the entire bundle down into the uterine lining. And sooner or later, the mother will notice.
Even brush.
One milestone event takes place just 2 weeks after conception, when the blastocyst is about the size of a poppy seed. This is the moment when the cells start to organize themselves into an embryo. The process is called gastrulation. With animals like frogs, whose embryos develop inside transparent eggs, it's easy to see it in action. After the egg becomes a hollow ball of many cells, some cells dive into the center, forming layers, which will go on to develop into different organs. In humans, gastrulation happens deep inside the mother's uterine lining, so it can't be photographed. But we think it works something like this. The blastocyst creates two oblong bubbles, one on top of the other. Sandwiched between them is a thin layer of cells. These are the cells which one day may become a baby. At the beginning of gastrulation, some cells begin moving toward the center. Then they dive downwards, creating a new lower layer. More cells plunge through, squeezing in between, forming a third. The cells in the three layers may not look different, but for each layer, a very different future lies ahead. The lower cells are destined to form structures like the lungs, liver, and the lining of the digestive tract. The middle layer will form the heart, muscles, bones, and blood. And the top layer will create the nervous system, including the spinal cord and the brain, as well as an outer covering of skin and eventually hair. This is a human embryo 3 weeks after fertilization. Less than 1/10 of an inch long, its neural tube, the beginning of the nervous system, is already in place. A couple of days later, the top of the tube is bulging outwards on its way to becoming a brain. With the primitive brain cells exposed, we can see some are sending feelers, making connections to their neighbors. As the days pass, changes proceed at a rapid-fire pace throughout the embryo. Everywhere, cells are multiplying, and they're on the move. Some reach out to one another, forming blood vessels. A heart begins to beat. As the embryo lengthens, the precursor to the backbone forms. Groups of cells bulge out on the sides, the beginnings of arms and legs. This is the embryo 4.5 weeks after fertilization. It is only about 1/5 of an inch long. The primitive backbone now curls into a tail, which will disappear in a few weeks. A large brain is developing, and on the side of the head, an eye. How does this happen? How does the embryo transform itself from a blob of cells into different tissues and organs, and finally into a fully functional baby?